Thank you all for joining us today. Please note that you are not audible or visible, but we encourage you to ask questions through the Q&A function on Zoom. This session is being recorded and will be available at the Hastings Center's website later today, and the link to the special report and the Community Sounding Board article will be placed in the chat shortly. We are excited to have many of the working group members join us for the event today. And first, I would like to turn it over to Erik, Parens, and Meyer.
Welcome, everyone. On behalf of my co-PI, Meyer, the members of our working group who are available today, and myself. The primary goal of this event is to entice all of you to read our just published report, which comes out of our 3-year project called Wrestling with Social and Behavioral Genomics, Risks, Potential Benefits, and Ethical Responsibility. At best, today we can scratch the surface of the rich content that you can find in the report. If we're successful, you'll become intrigued enough to read the report and do some of your own wrestling. Now, this slide shows the table of contents for the special issue of the Hastings Center report that grows out of our project. The first entry is our main consensus report, and the second entry is an article that describes our experience creating a community sounding board. Although Michelle and I need to focus our comments on the main report, our fellow steering committee member, Daphne Marchenko, who chaired the community sounding board, would be happy to address questions in the Q&A. The main report and article, as Ashman just said, about the article about the community sounding board, as well as some additional resources, are available at the link in the chat or at the Hastings Center's website. With the phrase social and behavioral genomics, or SBG for short, we are referring to research that uses genomic information to study social and behavioral phenotypes, where a phenotype is virtually any observable or measurable trait or outcome. SBG researchers study everything from obesity and substance use to subjective well-being and educational attainment. Educational attainment, which refers to the number of years of school one has completed, served as the running example in our project report. This slide features the 19 members of our working group. We are deeply proud of the demographic, disciplinary, and perspectival diversity of our group. Because our working group included historians of eugenics and scientific racism, our group was exquisitely attentive to the historical context out of which SBG research has grown, and our group was equally attentive to our current social context, where white supremacists and others are standing by, hoping for any genetic morsel that sounds to them like it can be used to shore up their agenda. Because our working group included bioethicists, we were exquisitely attentive to the additional ethical and social risks associated with SBG research, including the risk of exacerbating stigma and discrimination against individuals and groups, the risk of distracting policymakers from more effective ways of understanding or addressing the phenomena investigated by such research, and the risk of promoting the deeply erroneous view that the status quo is inevitable or that environmental interventions are futile. And because our working group included geneticists and social scientists who are currently doing SBG research, we were equally attuned to the potential benefits of and motivations behind such research, including improving social science research by using genomic data as control variables, improving health research by factoring genomic data about social phenotypes into making predictions about healthy outcomes, and helping disprove genetic determinism by showing the limits of genetic influences on social and behavioral outcomes. The research tool that our working group focused on, the polygenic index or polygenic risk score or polygenic score, The PGI is used ubiquitously by geneticists today, whether they're studying medical, behavioral, or social phenotypes. As many of you know, a PGI is simply a number equal to the sum of the magnitudes of the associations between single nucleotide polymorphisms and a phenotype. It is a number that predicts someone's odds of exhibiting a phenotype. Throughout our working group discussions, we sought a middle way between exaggerating how little or how much PGIs can tell us. In our effort to avoid exaggerating how much PGIs can tell us, we emphasize that PGIs are built on correlations. PGIs can be used to make predictions about future outcomes, but even when within-family analyses point to the causal effects of genes, PGIs cannot by themselves reveal causal mechanisms. Moreover, PGIs are part of a probabilistic, not deterministic picture. Among other things, these limitations mean that we should be skeptical that so-called precision education, where educational interventions are tailored to individual children's genomes, we should be skeptical that precision education will arrive imminently or perhaps ever. But neither are PGIs as useless as some critics sometimes suggest. From another angle, we can see how the correlations that PGIs sum up can be useful for doing better social science research. To use our report's running example, the fourth and largest of the educational attainment studies reported that approximately 15% of the total variation with respect to educational attainment among people of European ancestries, about 15% of the total could be attributed to correlations between common SNPs and educational attainment. That is, PGIs can be useful to social science researchers seeking to understand how myriad variables, both genetic and environmental, contribute to an outcome as complex as educational attainment. Although accounting for 15% of the total variation might not sound terribly useful to people who do not do social science research, it is of considerable use to people who do such research. For perspective, the obviously important non-genetic variable that is combined parents' educational attainment accounts for about 19% of the observed variation, which is only 4% more than the genetic variable that is the PGI for the most recent educational attainment study, that figure of 15% that I mentioned earlier. So serving as a control variable in social science research is a far cry from the dystopian vision of using PGIs to identify infants destined or determined to have low or high educational attainment. As we were wrestling with what PGIs mean and what risks and benefits they entail, we also had to ongoingly wrestle with the scientific concept of genetic ancestral populations. We had to do that because, for reasons we can discuss later, the SNPs used to create PGIs are detected by genome-wide association studies, which depend upon the identification of genetic ancestral populations. Unfortunately, although genetic ancestral population is a scientific concept, it is not nearly as clear or distinct as scientists would wish. Moreover, this scientific concept is distressingly easy to conflate with the social concept of race or ethnicity. Nonetheless, today, the concept of a genetic ancestral population is foundational for doing genome-wide association studies and for creating PGIs, whether the phenotype being studied is medical, behavioral, or social. So at least for today, PGIs are specific to genetic ancestral populations. That is, it is not possible to take a PGI created to make predictions about members of one genetic ancestral group and generalize or port that PGI to try to make predictions about members of another genetic ancestral population, which is why, for example, the All of Us project is so committed to diversifying biobanks and correcting the current situation where most genome-wide association studies are done on people of European genetic ancestries. In addition to being non-generalizable or non-portable, at least for today, PGIs created in the study of one genetic ancestral population cannot legitimately be used to make comparisons between genetic ancestral populations. To the extent that white supremacists hope to shore up their belief with scientific facts, the scientific infeasibility of group comparisons can be a source of solace. But our working group chose not to take too much solace in the fact that today it is not scientifically feasible to use PGIs to compare genetic ancestral populations. Rather, we chose to address the question, what if someday it were possible to compare genetic ancestral populations with respect to a phenotype as sensitive as educational attainment? Although we did not all agree on the answer to that specific question, we did agree about an awful lot, in particular on what is required to engage in the responsible conduct and communication of SBG research in general. So now I will turn it over to Michelle, who will describe in more detail where our working group could and could not agree. Over to you, Michelle.
Thanks, Erik. So our assessment of the risks and potential benefits of SBG research led us to conclude that SBG research on a wide range of phenotypes can be worth conducting, funding, and publishing. At the same time, we agreed that the social risks of this research, beyond what IRBs typically consider, mean that researchers, funders, editors, and journalists have enhanced ethical obligations to anticipate and take measures to mitigate these risks. In particular, we articulate two levels of concern, and we offer associated recommendations for each. First, SBG research of heightened concern is that which involves a sensitive phenotype. There is no perfect definition of sensitivity, and sensitivity can vary according to time and place. But a phenotype is generally sensitive if, for instance, it's very consequential to social status, such as educational attainment. It's part of a stereotype about groups, such as financial prowess, that in the context of genomic research can contribute to the erroneous and harmful belief that social identities are biological, or it's central to a minoritized group's identity, such as sexual behavior. We concluded that heightened obligations of responsible conduct and communication apply to conducting, funding, and publishing this type of SBG research. We can't, of course, offer an exhaustive account of what responsible conduct and communication of SBG research involving sensitive phenotypes entails. Among other reasons, that will have to be study specific. Nor do I have time even to go through all the examples we did discuss in the paper, which you can see listed here. And I should say figure 4, of course, appears in the paper, and you can peruse it at perhaps a higher resolution at your leisure. But I'll highlight just a few. So responsible conduct and communication includes, for instance, engaging people with the most at stake in the proposed research, justifying the use and definition of population groupings as inclusion and exclusion criteria, and moving away from language to describe populations that can easily be conflated with race or ethnicity, reporting effect sizes in the abstract and avoiding exaggerating them, including in graphs, and diverting misinterpretations and misuses of the research by clarifying both in the journal article and outside of it, for instance, in videos, press releases, and frequently asked questions, what the study does and doesn't mean, and how its results should and should not be used. Of course, some of these things, like adequate power and replication and independent samples, are good ideas or, in fact, even ethically required for any SBG research and indeed for any empirical research. Second, SBG research of greatest concern is that which involves a sensitive phenotype and attempts to compare groups identified by race, ethnicity, or genetic ancestry where genetic ancestry could easily be misunderstood as race or ethnicity, as is the case, for instance, with continental level genetic ancestries. We concluded that conducting, funding, or publishing this kind of SVG research requires a compelling justification. We all agree that this compelling justification at least requires a convincing argument that the results of this kind of group comparison research would be scientifically meaningful, because our default position is that they would instead be profoundly confounded by different allele frequencies and linkage to disequilibrium patterns, and by the different environments in which groups live, even within a geographic area, that shape behavioral and social phenotypes. And we all agree that, considering the social risks of group comparison research, scientific validity is an ethical precondition of conducting, funding, or publishing it. But as Erik said, although we disagree about how likely this is, none of us can rule out the possibility that group comparison research might one day be scientifically valid. And in that case, we disagree about whether additional compelling justification is required. For some working group members, the answer is no. For them, responsibly pursuing and communicating robust scientific findings about the world, including findings about any human group differences, is an important freedom, and attempts to decide which scientific questions can and cannot be asked and answered may actually do more harm than good. For other working group members, scientific freedom is only one value that must be balanced against others. For them, group comparison research must not only be scientifically valid, it must also have a favorable risk-benefit profile. Members differ in how likely they think it is that a group comparison study could meet this test, and which factors tend to suggest a more favorable risk-benefit profile. Possibilities include a study that compares non-minoritized groups of relatively equal power, and a study where members of both groups being compared support the research. Needless to say, we have not provided an algorithm for deciding which social and behavioral phenotypes are sensitive, nor an exhaustive accounting of how to responsibly conduct and communicate such studies. As for the SVG research we agree is of greatest concern, attempts to compare groups according to sensitive phenotypes, we are split, other than agreeing that scientific validity is essential and, at present, very unlikely. At one point in the project, Erik and I anticipated that this project would result in a series of essays by individual working group members instead of this consensus report. But working group members themselves recommended that we instead try to say something together. Although essays written by individuals free from the constraints of compromise surely would have been able to reach stronger, more specific conclusions, we hope that there's value in describing where diverse scholars can agree regarding this field of research, and that the consensus we were able to achieve is all the more meaningful for that diversity. As Erik noted, he and I have only briefly scratched the surface of a few main themes of our report. We are delighted that so many members of the working group made the time to be here today to help us flesh out those themes and to provide their own individual perspectives on SVG research in response to your questions. And we will turn to that now.
Great. Thank you, Michelle. I want to encourage people in the audience to please put your questions in the Q&A. One question so far is about benefits. And would someone on our panel be willing to say a bit more about what benefits such research might entail? Any takers?
I'm happy to say something, Erik.
Oh, great. Great, Patrick.
So I first, just before I say this, just want to share a little bit how meaningful being part of this working group has been for me. I've been able to have the chance to interact with a lot of people that I wouldn't necessarily be interacting with in a professional capacity. And I learned a lot. It changed the way that I think about a lot of the work that I do. And even a lot of the work, it's changed the way that I do the work that I do. Just as a background, I'm an economist with a background in doing genetics. So I do social science genetics research. And one area of research that I've done that I think has been valuable that wasn't touched on as much in the previous comments was thinking about heterogeneity. I think that we do a lot of work where we just look at average effects. Does this policy affect health on average? Does this policy help well-being on average across the population? Often it's important to us to know who is being most and least affected by these policies as we think about inequality and health disparities in society. And so, for example, I wrote a paper with a couple of my colleagues at USC where we looked at an education reform in the UK, and which increased education, increased the compulsory schooling age laws in the UK, which increased education in the UK, and subsequently saw an average increase in health in the UK, specifically measured by obesity risk. And when we were able to split our sample by people who are at high risk of obesity based on their genetic predictions versus low risk of obesity, we found that people who are at the highest risk of obesity benefited most to these educational reforms. And so I think that knowing not only that education improves health, but decreases health disparities is an important lesson that we learned from this research. And this is the kind of question I think that using genetic data helps us understand that can really be beneficial.
Thanks, Patrick. Since you mentioned what you learned as a scientist participating in this diverse group, I wonder if any of the non-scientists might like to comment on what they learned by participating in the project. Maybe Jim Tabry, would you be willing to say a few words?
Sure, happy to. Like Patrick, I learned a great deal from this group. I'm a philosopher by training. And much of my familiarity with research on things like the genetics of educational attainment or poverty was through the lens of high-profile people looking for the genes that did that stuff, with talk of things like precision education or personalized education. And so I think I had a certain view about what this scientific research looked like. And so it was very eye-opening for me to interact with this sophisticated group of scientists who brought our attention to all the various ways that you might be interested in this research that really have nothing to do with trying to find the gene for intelligence, about better study design, about controlling for factors that allow for better social science research. That was something that was eye-opening for me, and I learned a lot from hearing the different perspectives there.
It looks like Dan Benjamin wanted to add. Did I see something, a thought occurring to you, Evelyn?
Yes. Sure. I actually thought that I don't necessarily think of myself as a non-scientist, but certainly a non-geneticist or genomicist. And I have to say, I mean, I study the history of genetics and the history of biology and the life sciences and medicine. So I think it was really important for me to hear directly from people doing the work instead of just reading their published papers about how they construct and think about the questions that are raised by the PGI research, and also just generally socio-behavioral social science. Because here directly, I began to see ways in which we thought similarly about some things and certainly very differently about other things, but I didn't have the specificity until we had those conversations. So that is one of the important things I think I got out of the conversations that we've had over, or I should say our wrestling over the last few years.
Thanks. Dan, did you want to jump in?
Sure.
On the benefits question, or?
Sure. Well, I mean, I'll also just briefly say that, I also learned an enormous amount from everyone on the panel, and about how, especially about how the things that we write and, are kind of standard approaches in the research are so easily misunderstood, not just by the public, which I think I was aware of before, but also by people who are just not specialists in socio-behavioral genomics. And so it's just, so important to be careful about things like the way that we describe genetic ancestry and be accurate about it and clear about what we mean. On the benefits, I just wanted to add, and this is something that Jim mentioned as well. I think one of the most important benefits is controlling for genetic factors when studying environmental effects. So for example, if you want to study the effect of providing preschool, then your ability to, you can do a randomized experiment where you randomly give preschool to some kids and then see how that affects their academic achievement. But those experiments are very expensive to conduct. So any control variables you can use to improve the precision of your estimates is extremely valuable. And genetic data can be very useful that way. And I think there's many such examples like that where genetic data could be useful across the social sciences for questions that themselves have nothing to do with genetics.
Thanks, Dan.
Erik Turkheimer? Yeah, thank you, Dan. We're going to jump in, Erik Turkheimer.
Hi, I hope everybody can hear me. Unfortunately, I've wound up traveling during the meeting. This has been such an important group to be a part of, and it's meant a great deal to me. I wanted to say something about the question about the difference between biological and social phenotypes, because there are important differences between them that are scientific differences. They're not just ethical or philosophical differences. When we talk about human beings and human genetics as a biological thing, that science can be based on commonalities that we have with lower organisms that can be studied in experimental ways that are not available in human beings. So if we're trying to understand the genetics of kidney function, other mammals provides a very powerful basis for what we can know about humans. But when we study the relationship between genetics and educational attainment, we don't have that. And that means we have to rely on what we can infer from the way human beings naturally vary in educational attainment without being able to do experiments, without being able to assign people to different kinds of environmental conditions to control for the obvious environmental effects on things like educational attainment. So that means that the science about educational attainment, as hard as we try, can never be as certain as the science about biological characteristics. And that uncertainty constrains the ethics of what we can do and what we can conclude. So I think it's very important to realize that the science underlying biology and social characteristics are not the same. And that places different constraints on what we can do and how we interpret what we find. I'll mute myself again.
Thanks, Erik.
Dalton, could I ask you to perhaps weigh in on what motivates you to participate in or to conduct this research and what you see as the benefits?
Sure. Thanks, Michelle. Just in addition to what others have already said about the benefits of the use of polygenic indices for educational attainment or for any social behavioral trait to increase our understanding of heterogeneous effects and distributional effects, as Patrick was mentioning, or to increase our statistical power in just knowing whether even something like a randomized trial has a valuable impact or not. I find it useful to use genetics as a tool to do purely social science as well, as a tool that we didn't have 15 years ago. So because of the sort of the magic of meiosis, when we have a child, when we have parents' genotypes, and we essentially hold those constant and look at the outcome of a child in a family and measure their genotype, it's really a random shuffle of the parents' genes. So we get this kind of this experiment with each child born. And that allows us, if we measure the genotypes of everybody in a family, to really understand social dynamics in a family, for example. So we can understand how different aspects of a child affect the rest of the family. We can understand, for example, if it does a child with a high propensity toward ADHD or toward depression, how does that affect the mood of the parents or of the siblings of the family? We couldn't ask or answer that kind of question before we had these kind of data or measures. And we can extend that to understanding a whole range of social phenomenon, like peer effects in high school on smoking, something that we've looked at, and any number of other outcomes, because genetics, and specifically with respect to educational attainment, we know it is predictive, and we know that within families, at least, it's kind of a pure effect. And it, again, without trying to argue for nature or nurture, we can just use that as a tool to understand social transmission and environmental effects in groups or in families in particular. So I find that it's opened up a whole new sort of area of inquiry in the social sciences that wasn't possible before.
Michelle, I noticed that there is sort of another line of questioning that maybe we can take up sooner rather than later. One person asks, I have a background in the life sciences and am a social scientist who works on science policy in the criminal legal system. I have a difficult time seeing how SBG can benefit the criminal process. There are already so many unfounded narratives about criminality and dangerousness in play without the use of SBG. Do any working group members think it's possible for SBG to be used in the criminal process? And the rest of the message disappeared for me, but you get the gist of it. Is anybody?
Well, Daphne, I think, Erik, you mentioned the community sounding board, and my
Yep.
recollection is that this was a particular concern of theirs. So Daphne,
Yep.
would you invite you to say something about that?
I'd be happy to. So there was a community sounding board that was formed alongside the academic working group, comprised of members of the public. And you can read through that process paper to try and get further information on our reasoning for building out a community sounding board and some of the lessons learned from that experience. But Michelle's right that a big concern of a number of community sounding board members, many of whom were members of Black and Brown communities, racialized minority communities, was of the potential use of social and behavioral genomic data by law enforcement and in the criminal justice system. So I would just, I think, echo that person's concern about the use of SBG research in criminal justice system, and add that many of the members of our community sounding board saw social and behavioral genomic research on criminal and aggressive behaviors to be of most concern for them, and in particular, most concern because of the ways in which that data might be used by members of law enforcement and the criminal justice system, which, as I'm sure many here in the United States know, has a number of challenges around systemic racism and racial injustice.
Paul, I wonder if you have some additional thoughts about that line questioning.
Yeah, so the primary purpose of the criminal justice system is adjudicating guilt and innocence. And it would not appear that SBG data would have any benefit in that process. However, we have some recent experience with the use of more primitive socio-behavioral genetic data, namely the MAOA gene, which was said to be associated with aggressive behavior in early studies in the criminal justice system in the sentencing phase of the process. That experience, I think, taught us two things relevant to this question. One is one needs to be extraordinarily careful about how such information is used, because as time went on, it became less and less clear that the MAOA gene, actually the allele that was said to be associated with violent behavior, actually was. And despite the credentials of the original proposers of that relationship, it seems quite uncertain and unlikely to be true at this point. But the second thing is that that effort was not terribly effective. And that's been shown not only by the real life cases in which it was used, but also in a large number by now of experimental studies where it seemed to make little or no difference to people's sentencing decisions. And we might actually credit the general public, the people who serve on juries, and judges who most often engage in sentencing decisions, with a good deal of wisdom on this, recognizing that some skepticism is required when looking at data of this sort. And also that predictions of future behavior are inherently uncertain and contingent and not dependent exclusively or even primarily on genetic factors, and declining to take them into account in their decisions. And so the bottom line is it's very difficult to see any use of socio-behavioral genomic data in the criminal justice process that could at all be justifiable.
There are, I'm seeing a couple of terrific questions. One of them I'm not absolutely certain I understand, but I will try to translate it, and if I've misunderstood, the person who wrote it can clarify. This person says, per the presenters, the generation of GWAS plus PRS for E and EA involves both genetic ancestry groups and socially sensitive trait. Could EA GWAS, could an educational attainment GWAS itself be subject to unjustifiability analysis, not only further downstream uses, such as further comparisons between genetic ancestry groups? I think the person is asking, did we ask the question, is the EA GWAS itself justifiable or not? And I think if that's the question, I think we owe it to you to give a clear answer. We take EA to be a sensitive phenotype, but insofar as it is done within one ancestral group, if it is conducted responsibly and communicated about reasonably, then we wouldn't take issue with it. If I misunderstood the question, please restate it.
Erik, the questioner actually has clarified that that was in fact what they were asking. And so, great, you've now answered it. There was unless you have more on that, I was going to move to a different question. Okay, there was a question specifically for Erik with a C, Erik Turkheimer, about some remarks that you had said before your analogy to a CD. Do you want to read that question and tackle it? First, you need to unmute, though.
Yes. Let me switch to chat so I can read it. Or I think I can paraphrase it, probably easier. I cited an example a long time ago, talking about why, in my opinion, it was unlikely that we were going to come up with genetic explanations of very complex human behaviors by drawing an analogy to a DVD or a CD, which for the young people out there was a medium we used to use to watch movies back in the day. And imagine a situation where somebody was peering at DVDs through a microscope to look at the little pattern of laser dots on the microscope and had been given the task of figuring out on that basis whether the DVD in question was a comedy or a drama in the movie, and suggested that no matter how good we were with those microscopes, we would never be able to do that because the complex content of the movie didn't translate down to that level. And the question was, I do, but I think there's an important caveat there that goes back to what Dalton Conley was saying a few minutes ago. I think there are two different sorts of things you can see happening on the basis of sociogenomic GWAS. One is, and I think this was once upon a time an explanation, that we were going to use GWAS to understand why the mechanism that explained why some people got more education than others. And we were going to take all of these formerly psychological things and boil them down to a biological level and have biological explanations of our psychology that we hadn't had before. That for almost 100% has not happened. I think it's very unlikely to ever happen. And we shouldn't base our expectations on the likelihood that that's going to happen. On the other hand, as Dalton said, without doing any of that, PGIs and GWAS generally can provide us with useful tools that we can use around the edges to understand complex social things like educational attainment that are very hard to study scientifically from the get-go for the reasons I mentioned before, that we can't randomly assign people to educational conditions or anything like that. And those tools have allowed us to do social science a little bit better than we were able to do it before without sort of terrifying if they ever happened, but I still don't think they ever will, so they're not really worth worrying about.
I'd like to add on to a point I think Erik.
I hope that's just a question.
Yeah, I think you made a great point there, Erik. I think one thing that we've come to learn about PGIs is that they're, as a summative index, is that it's important to take a step back and think about what we're also learning about polygenic effects, meaning that from most of these genome-wide studies, we see that many of the diseases or the traits that we're studying share a lot in common with other diseases. We'll refer to it as pleiotropy or genetic correlation. And so to say that there's a polygenic index for X being distinct from a polygenic index for Y without necessarily understanding why that is something that anyone who wants to use a polygenic index needs to consider before they apply that index. And so thinking about the fact that traits don't occur in a vacuum. And in humans, there are social interactions, multiple aspects about who we are, play into our decision-making, our behaviors, is certainly something that we need to, bring into how we model behaviors, or design studies, whether we're using a single polygenic score or multiple polygenic scores, or studying multiple behaviors, which is what we see oftentimes when looking at these complex social behavioral outcomes.
Can I add one small thing as well? Just to tie this to a question that someone asked earlier about, how we can distinguish between whether something is social or biological. And I think that one thing that we've learned is that the boundaries are not clear, and that, genes, especially for the complex traits that are studied in social science genomics research, they operate through the environment in almost every case. And so, I don't know that we can easily talk about whether a thing is biological or environmental. That can't be, it seems like that can't play into any sort of criteria as we're thinking about this kind of research, because everything is a bit fuzzy. I think that, and I don't think in the paper that we released, we do that very much. I think that we talk about phenotypes that are sensitive, and studies that are of greatest concern. But I think that we didn't attempt to try to slice and dice different phenotypes into, this one is biology and this one is environment and this one is 50-50 or whatever, because just everything is all meshed together.
Could we pivot again from this very important question about the relationship between the biological and the social to this question about privacy? Is there anybody who would like to address that one? Michelle?
Give me a minute to process it, Erik. In the meantime, Daphne, do you want to? We've had lots of questions, not surprisingly, about what I think our group might call downstream applications of the basic science. Do you want to say a bit more about that?
Yeah, so, I think initially when the working group was formed, thinking about downstream implications was perhaps not an initial central focus of the group, but we quickly realized that that was a central facet to considering risk, potential benefits, and ethical responsibilities. And I would also say, at least in my personal opinion, some of the work that was done with the community sounding board, in which we're bringing together members of the public who are seeing the downstream implications or wanting to think through the downstream implications because it's most immediately relevant to them, helped, I think, steer some of our thinking. So, one example, which we talk about in the process paper for the community sounding board, was that sounding board members were very interested in the potential applications of social and behavioral genomic data by members of industry, and were in particular concerned about these potential industry applications of the data. And having that conversation with members of the community sounding board signaled for the subcommittee, so that the subcommittee was comprised of Erik, Michelle, Patrick, Nanaba, Garrison, Shaniqua Collier, and Sandra Lee, trying to find ways to bring that conversation that was happening among community sounding board members about industry uses of the data to some of the conversations that were happening with the larger academic working group. So, I do think that it can be very difficult and challenging to think about and anticipate potential downstream implications of social and behavioral genomic data. I think we were seeing in the community sounding board, members of the public were very interested in those potential implications, given the feeling that those applications would be most immediately felt or experienced by them. And so that kind of informed some of the thinking that the academic working group was doing.
Thanks, Daphne. And I'll circle around to the question that Erik Parens had mentioned, which is, so this is a careful reader, thank you so much, who has also already found box 3 of our report. And this is a box where we describe a handful of potential downstream applications of PGIs. And as we say in the report, we certainly don't agree on whether any of those potential downstream applications and practice or policy, like in the criminal justice system or in the education system, et cetera, would be good or not. And, we were limited in our ability to thoroughly analyze any of those particular use cases. We were really focusing on the basic science, understanding what PGIs can and can't tell us, their general risks and benefits, et cetera. As an applied ethicist myself, my own personal view is that you would need to staff a committee like this with people who have expertise in a particular domain. So I guess I would be skeptical about one group who is able to comprehensively tackle the ethics of many, many different domains like that. all of that said, the question is about the particular use case of using PGIs in the education system, something like a offering extra money for people with lower PGIs for educational attainment or for learning disabilities, different phenotypes, for example, offering schools more resources or individual students more resources. And the questioner is rightly noting that privacy concerns, and lots of potential concerns with that application, one of which is the idea that this is, potentially sensitive information that would be shared necessarily with the administrators and the folks implementing such a program. And so the question is, did we consider the privacy risks? And the short answer is no, only in the sense that we didn't, as I said, comprehensively dissect or analyze any particular use case, except to say, here are some of the things on the horizon, and we have to be aware of those and take those into account when we think about the risks and potential benefits, I suppose, of the basic science, thinking about how it could be used or misused downstream. And I suspect that many, if not all of us, would agree that an application like that certainly implicates privacy, and also, as the questioner also suggests, has the ability to be used contrary to the interests of the people it's meant to help. Dan, Benjamin, I think there's a question about causation that perhaps you could weigh in on.
Sure. So someone raised the question of whether in the report we talked about we made a distinction between using genetic data to predict behavior and being able to understand biological causation. And I actually think there's kind of two parts to that. One is biological, and the other is causation. They're not exactly the same issue. And I think the short answer is yes. There's a lot of discussion in the report about how one of the major limitations of current PGIs is that they are merely predictors. But there's two subtleties and caveats to that observation. One is that how much that's really a limitation depends on the application that you want to use the PGIs for. So in the example I gave before about controlling for genetic factors, when you want to look at the effect of preschool, is if you have a randomized experiment to evaluate the effect of preschool, you don't need a causal variable as a control. A quote mere predictor is perfectly adequate as a control that increases your ability to detect the effect of preschool. The other subtlety, something we had to be attentive to when writing the report, is that the frontier of knowledge is advancing quickly. And although right now PGIs are based on correlations, there's a growing amount of family data that allows for within family studies, like looking at the genetic differences between siblings and seeing how that relates to their social and behavioral phenotypes. And that lets you say something more confident about causal effects of the genetic variants. There's also, in some cases, more knowledge about the biological mechanisms by which at least some of the associations, in some cases, might be the mechanism. And so those considerations kept us from having blanket statements about the just genetics being predictive and not having any causal knowledge of causal mechanisms, and led us to formulate recommendations that were focused on general principles for determining, for evaluating research.
Thanks, Dan. We have a question, maybe a couple of questions about the portability problem. Rowan, I was wondering if you might be able to speak to that.
Ron, can't hear you.
Thanks. After 3 years. So I think partially the question says today's BGIs can't be ported. There seem to be a large number of papers and commercial products that do seem to port these, even with some attenuation to predictive power. And why do we say that they cannot be? I think partly one of the reasons why we say they cannot be, but also provide recommendations for why they shouldn't be, sort of within the report. And in large part, we allude to differences in linkage disequilibrium, as well as, which refers to allele frequencies and haplotype blocks in which the alleles are inherited across populations. And while there are maps that can be used to try to essentially sort of infer what the probable effect sizes would be in those populations, it is essentially a guess, a best guess. And Dr. Martin, I'm sure, could speak more to about that once I'm done here. But even so, some of the things that we've seen in our own studies is that you might make those inferences, and the types of methods or the assumptions you have to make in those models for how you create those PGSs don't generalize very well from one population to the next. And so if you're making different assumptions, you're also making you're essentially making more assumptions about how these factors are operating differently across groups, when in fact you don't necessarily know how they are operating at all. So again, another reason why you shouldn't be trying to use them if they lack not just portability, it's more so they lack face validity in that regard, right? In the same way we did sort of design psychometric instruments, right? For instance, if you're looking at a construct like impulse control, and you design a survey to measure an aspect of impulse control, you wouldn't necessarily expect that to vary across different subgroups, right? In terms of the kinds of questions that you might ask to do that. And so you would test for that construct to be valid across different populations. And that's certainly something that one would need to do. The other aspect to this is the combination of genetic and environmental factors might also vary across groups as well. And it's the case that in many of these instances, the sample sizes have not been sufficiently large enough for us to be able to design robust studies in a way to explore those factors, or at least begin to account for them within how we apply these PGSs.
Thanks, Rowan. We definitely want to hear from Alicia on this point, but Evelyn has to run. So Evelyn, please share your parting comments with us before you do.
Okay. I guess I just wanted to say a word for the people here about the process we've been engaged in for 3 years. We're making sort of side comments every couple of minutes about being together for 3 years. And I want to say that it has been extraordinarily intellectually challenging, sometimes difficult, sometimes deeply enlightening. But I do think it's a process that allows for certain kinds of questions to be raised. And I can't think of other venues in which those questions could be raised. Some of the limitations down in the specifics of the kind of research and work that we all do, the need for thinking about how our work might be perceived and understood outside of our narrow lanes of our disciplinary research, and the implications it might have for those people who are completely outside of the academy and the research enterprise. I think it's important to think that at certain periods of time, we need to have this kind of process. It's not simple, it's not easy, and it takes a lot of time. But I do think at the end of the day, it's a very worthwhile exercise. It puts us all, I think, in a better place, both of understanding our research from our own perspective, but also the possible impact and influence that our work has on people outside of our fields and the broader public. I think that's an important thing to do at this particular point when we have people who are, the technology and the science we do is becoming more and more complex, but also more and more impactful. on broader communities. And I think us wrestling during this time is an important way for us to be able to speak more clearly, distinctly, and carefully about the risk and limitations of the work we do. So thanks a lot.
Thank you, Evelyn.
Thank you so much, Evelyn.
We're going to extend just a few more minutes because we have a few lingering questions and comments for those who are able to stay. So Alicia, please, let's pivot back to that. And we have two more Q&As and then we'll call it a day.
Sounds good. It's hard to follow Evelyn on that because it's been such a wonderful time working with all of you. Just coming back to the polygenic index portability question, there are a few ways that people can think of challenges to portability. One is in this sort of strict differences in accuracy point. So taking polygenic indices generated from one genetic ancestry group and trying to port them to another group, we know that there's a loss of accuracy if we have an unrepresentative or imbalanced study. That's still true for the vast majority of biomedical phenotypes and socio-behavioral phenotypes. There are a few exceptions to that where we're predicting with reasonably decent and consistent accuracy across different genetic ancestries, but by and large, we're still sort of on the upswing of genetic discovery, which means that there is still a gap in prediction accuracy among different genetic ancestry groups. Now, when thinking about this portability gap, you might also think in how these polygenic indices translate, and there's also a translation gap, and because of these differences in accuracy, as well as baseline risk for some biomedical diseases, for example. So if you're trying to implement a polygenic score, polygenic index, you might act differently in different genetic ancestries simply on the basis of their ancestry and the prediction accuracy. And so that's clearly a problem here. When thinking about policy changes on the basis of things that have differential accuracies. So some health outcome examples that you could think of include areas where there's differences in baseline risk, perhaps as a consequence of genetics, but also a huge environmental component. And then if you have a polygenic index that might actually put you over some threshold for getting some intervention or for some treatment, then having a polygenic index that's differentially predictive may mean that more people at high risk are being treated or fewer people who are at the highest risk are being treated. So I think we need to be very cautious about translation. And I think there's a lot more work that needs to be done on the implementation and science for polygenic indices generally. So this doesn't just reflect genetic ancestry differences. There's all sorts of differences that this group has explored with polygenic indices, Dalton, for example, and others in this call, but I'll leave it at that.
Thanks, Alicia. Erik, we have a question about disabilities. Would you mind taking that one?
Yeah, I do want to take that, and then I'll turn it over to you, Michelle. I'm so grateful to everybody in this group. It was really lovely, I thought, the way Evelyn summed up her own experience. I feel incredibly grateful to have participated in such a diverse group with respect to demographic differences, disciplinary differences, and perspectival differences. And we should pat ourselves on the back for that. And we should be grateful for the opportunity. And as we do that, I want to acknowledge that while we had myself as a longtime ally of the disability community on the group, we did not have a person who identifies as disabled on the group. This was a defect in our process. The next time somebody does a group like this, they will have to include representation of disabled people. It was a mistake. We were doing the best we could, juggling all sorts of diversity, and we failed on that front. And I just want to I want to say that it was a failure. Next time we will do better. Whoever does something like this will do better. I hope that despite that, there is lots to be learned. I mean, I know that there is lots to be learned from our process. Michelle, do you want to?
Well, I'd like to invite Nanaba to give some comments about the sounding board.
Thank you, Michelle. Yeah, so I was also one of the academic group members who participated in the sounding board conversations. And I just want to underscore the importance of bringing in community voices. It was so impactful and powerful for us to hear from people who had no or very little expertise in the science and behavioral genomics and the research that has been going on. So to hear their perspectives was really meaningful. So Daphne led a lot of that effort, and I just wanted to underscore. They raised questions about accessibility of these types of results. They raised questions about concerns about stigmatization and discrimination if these results were to be misused in different community settings. People talked about historical harms. And so it was really important for us to not lose sight of that voice. And so I think for any kind of research that touches on these types of topics, it's always important to include those voices as well. Thank you.
Thank you. Ashna, do you want to close us out? Thanks to those of you who have been willing to stick around for a few minutes over our allotted time.
Yes, thank you all for joining us today. As a reminder, a recording of this will be available shortly on the Hastings Center website, and a special online hub has been created on the Hastings Center's website with links to the report and additional resources. And you can find these links in the chat as well. Thank you all again for joining.